Hi, Assalamualaikum. My name is Nur Amali Shahira binti Abdul Hadi. Okay, I want to present for my individual assignment. The topic that I choose is breeding management. Okay, the fish breeding. The fish breeding is the producing the young ones from the parent broodfish and to ensure the species continuity and survival. In aquaculture, fish breeding is also very important activity, which is to ensuring the availability of fingerling for the stocking of pond, tank and cage. Also, the in reproduction of the breeding controlled by internal and external or environmental factor. The environmental factor include the temperature, salinity, good water quality, adequate water, also the photo period. For the biotechnology in fish breeding, we use the hormone, cryopreservation, which is fish sperm, coral cell, and fish embryo, and then we use the DNA marker, and then we use the chromosome manipulation, which is polyploidy, androgenesis, and genogenesis. And also we use the transgenic fish. For the use hormone of breeding, many of fish spawn in environment that are nearly impossible to stimulate in a hatchery. The induced spawning can be used to produce hybrid that are different from the parent species, and then produce the sterile polyploid fish can produce the fry outside the normal spawning season for maximum hatchery production, and also the maximize the survival of fry under the controlled hatchery condition. For the hormone for induced breeding, we use the pituitary homogenate, human chorionic gonadotrophin, HCG, gonadotrophin releasing hormone GnRH, luteinizing hormone releasing hormone LHRH, and synthetic hormone ovaprine. For the recombinant hormone is the growth hormone that can make a fish bigger and faster to the market, and also growth hormone could shorten the growth time needed for farm rice to reach the market size. And then the production of recombinant protein involve of cloning of cDNA. And for the hormone growth factor produced from a large variety of fish species currently used in aquaculture industry. So the main factor that can consider when producing and employing recombinant hormone are: first, cost-effective production method, efficient method for renaturation of recombinant, appropriate assay to determine their biological activity, and also the extension of half-life. For the cryopreservation, this is a process where cell or whole tissue are preserved by cooling to low sub-zero temperature, such as 77 Celsius until 196 Celsius at the boiling point of liquid nitrogen. At this low temperature, any the biological activity, including the biochemical reaction that lead to cell death, so we have to stop it. However, when cryoprotected solutions are not used, the cell being preserved and often damaged due to freezing during to approach to low temperature or warming to room temperature. The benefit of freezing is genotypic drift, transformation to tumor related to properties, contamination, distribution to others, 7 reaction, cross-contamination by other cell lines. So this is for DNA maker. We have the DNA maker for progeny identification. A gene of DNA sequence with a known location on a chromosome that can be used to identify individual or species, and also it can be described as a variation which is due to mutation or alteration. A progeny test is performed by mating the male with a number of female to produce many progeny in a different environment, and genetic marker have to be easily identified, associated with specific locus and highly polymorphic. Because of homozygote, detection of marker can be direct by RNA sequencing or indirect using enzymes. Here we have chromosome multiplication. The purpose of chromosome multiplication is higher growth rate, sterile and monocyte population of better yield. So the type of induction of chromosome multiplication, we have temperature shock, pressure shock, chemical exposure to ultraviolet. So the technique of chromosome manipulation, we have three, which is polyploidy, androgenesis and genogenesis. First, polyploidy, which is involve the addition in chromosome number. This is an organism or cell having more than twice the haploid number of chromosome. The type of polyploid, we have triploid, tetraploid, pentaploid, hexaploid, octaploid, decaploid, and dodecaploid. Second, we have androgenesis, which is limit inheritance to the paternal pair. Okay, the androgenesis, development of zygote that contain only paternal chromosome as after fertilization and oocyte one whose chromosomes are absent or inactive. This is also involve the development of an organism with only paternal pair or paternal chromosome, and also the androgenesis is induced by gamma or ultraviolet radiation of egg to inactive the chromosome DNA. Third, we have genogenesis, which is limit inheritance maternal parents. The genogenesis, the process of development of ovum after penetration of sperm, but the sperm is unable to form syngamy with nucleus of ovum, so genetic fish inherit genes only from the mother. So development of embryo contain only maternal chromosome because the egg is active by sperm without fusion of the egg and sperm nuclei. In some fish, the genogenesis is achieved naturally or artificially. And last, we have the transgenic fish. This is which is transgene has been artificially introduced by the transgene stably integrated into the genomes, and also transgenic fish produced that could benefit to the aquaculture industry, and the production of transgenic fish focus on the general application, which is to enhance traits of commercially important species, to develop fish as bioreactor to produce biomolecularly important protein, and to enhance the utility of fish as indicator of adverse health. That's all from me. Thank you.